This short lecture will introduce you to the Gram stain, a fundamental technique used in microbiology. Gram stain is one of the most important techniques used to identify bacteria. In fact, one of the first questions a microbiologist will ask is, what is the Gram stain? Is the organism you're telling me about gram-positive or gram-negative? The Gram stain is a differential stain. A differential stain is one that differentiates cellular components, and it can be used to distinguish between organisms. So think about that word, differential or differentiate. It's something that tells the difference between. And for the Gram stain, the cellular component that is differentiated is the cell wall. And the Gram stain differentiates bacteria into gram-positive and gram-negative groups. Gram-positive bacteria tend to be killed by antibiotics like penicillin. They act against the cell wall. And gram-negative bacteria are more resistant to antibiotics. Because the Gram stain procedure can be finicky, you must always use Gram stain controls. The Gram stain controls ensure that your Gram stain procedure was successful. And once you've made your Gram stain, you do want to look at the controls first when looking at this under the microscope, because if your Gram stain controls don't stain as expected, then you can't have confidence that your bacteria of interest stain properly. So what are the Gram stain controls? In Biology 207, we use Staph epidermidis as our gram-positive control. So Staph epidermidis, if done properly, is a cocci that stains purple. So gram-positive is purple. E. coli is our gram-negative control. E. coli is a bacilli, and it stains pink. So gram-negative bacteria are pink. So if these do not appear as expected, when you view them under the microscope, you need to prepare a new slide. So in making your slide, you typically have a minimum of 3 organisms on your slide. Your bacteria of interest, the gram-positive control, and the gram-negative control. Once your slide is prepared for staining, the first step in the Gram stain procedure is to add the first dye, crystal violet. And after each step in the procedure, water is used to rinse the slide. So you add the crystal violet, and after 45 seconds to a minute, you rinse off the crystal violet with water. After rinsing away the crystal violet, you add iodine. Iodine is a mordant, or a dye fixator, as Labster refers to it. The iodine complexes with the crystal violet to form a large molecule. After you rinse the iodine away, the next step is to decolorize with alcohol. The alcohol serves to dehydrate the thick peptidoglycan layer of the gram-positive bacteria that traps the crystal violet iodine molecule so it can't be rinsed away. In a gram-negative bacteria, that alcohol dissolves the outer membrane and the molecule is washed away, and the bacteria loses the stain. After another rinse with water, you add the counter stain, safranin. The safranin now stains the gram-negative bacteria a reddish pink color. And after a final water rinse, the bacteria are now ready for viewing under the microscope. So again, just a quick walkthrough. Crystal violet is the first stain used. That's followed by iodine, which is the mordant, which makes this complex. Alcohol is the decolorizing step. So alcohol will decolorize gram-negatives, allowing that crystal violet iodine complex to wash away. And then finally, the final stain, the counter stain, is safranin that will stain the gram-negative bacteria pink. For bacteria, the cell walls usually come in 2 types, gram-positive and gram-negative. Here's some electron micrographs of those cell walls. Your Labster simulation will go into more depth about the cell wall differences.